thank you so much. It's exciting to be part of this lecture series where we've heard from a lot of terrific people, and you've heard from a number of protease experts, and I consider myself one amongst their group. So in fact, I would like to advertise to other people who are interested in proteases that I'm co-chairing the Protease GRC this summer in Italy. We actually have 2 slots left for late-breaking talks, so please reach out to me by email if you have a story that you'd like to tell the protease community. So today I'd like to tell you about the work that's going on in my lab on antiviral projects. One is the one that I'll focus on mostly today, which is about a Zika virus protease inhibitor and about how this inhibitor impacts the dynamics of the cofactor region. And then I won't have time to get into these in detail, but my lab is also involved in developing inhibitors for chikungunya virus protease. And we also have developed a SARS-CoV-2 protease reporter that I think could address some of the issues that Aaron brought up in his talk. So maybe if there's extra time during the question and answers, we can also talk about those topics. For this audience, I don't think we need an introduction to flaviviruses. It's very clear to probably everyone on this call that flaviviral infections are a real problem. Dengue, Zika, yellow fever, West Nile, etc., are all problems across the world. But dengue itself has 390 million cases per year. And there aren't any antivirals available for this class of viruses, but we know that antivirals are essential even if there is a vaccine available. And just as a reminder, the reason why flavivirus proteases are excellent targets is because many viruses, including the flaviviruses, express a polyprotein, so a single protein chain that needs to be cleaved where you see all these vertical lines by a protease in order to get the active components of the virus, both the structural and the non-structural proteins. So in the flaviviruses, they have a protease that's composed of 2 domains. It's a 2-component system that has a protease core. It's a serine protease that has a chymotrypsin-like fold, and it has this cofactor, which is the NS2B region. The question we had in my lab was, can we make a selective inhibitor for one flaviviral protease, or might it be possible to make a pan-flaviviral protease inhibitor? So when we joined this field, we found that when we looked across the proteases from these different flaviviruses, they all had very different biochemical properties, but one of them stood out. So when you look at Zika virus protease, we see that it's much more active than all the rest of the proteases. Now, I described to you on the last slide how the protein itself is composed of a core. So here you see the core. You can see an active site inhibitor bound in the core. You can hear, and right here are where the catalytic residues are. And there's also this cofactor. And when we did a sequence alignment, we noticed that that cofactor is the most variant region of this family of proteases. So when we look at Zika virus protease, it's very active, much more active than dengue. But Maureen Hill asked the question, if we take the cofactor from dengue, that's why it's blue here, and we substitute that into our Zika virus protease, what happens to activity? And we found that that dengue cofactor, just this purple region here, knocks down activity a lot. And in fact, what I'm going to tell you in my talk today is that if we just take a teeny tiny region of that cofactor from dengue and put it in Zika, that really impacts activity. So we thought this was really interesting. And it was interesting because these proteases can adopt an open and closed conformation of this cofactor region. So in the open conformation, we see that this cofactor is folded down around the core of the protein. And conversely, that cofactor can also be in an up position. So our lab wanted to ask about the relationship between these open and closed conformations. So we introduced some cysteines into dengue virus protease, which doesn't have any native cysteines. And we introduced cysteine pairs that were compatible with the open conformation, but not the closed. We called that the open trap. We made one that was an ether trap, so we could form the disulfide in either conformation. And we made one that was a closed trap, so that it could only form in the closed conformation. And this analysis showed that the closed is the inactive conformation. So you actually need this cofactor to wrap up around the core of the protein in order to achieve an active enzyme. So what we asked is, can we control this open and closed ratio? And does this cofactor and its conformational change perhaps explain the substrate selectivity and the altered pathology that's observed in Zika virus protease? So at this point, we teamed up with Jim Wells and Olivier Julien, who are at UCSF and University of Alberta, and we scoured the entire human proteome. So we did a proteome-wide entomomics analysis, which allowed us to, excuse me, which allowed us to look at the substrates, the human substrates of Zika virus protease. And what we found is that just as would be predicted from the sequence of the polyprotein and looking at the cleavage sites, when cleaving human proteins, Zika virus proteases cleaves a sequence that is highly positively charged. So having G, K, R is what we saw in most of these cleaved sequences. So we saw 31 human protease substrates that had not been observed before. And I'm going to tell you today about 4 of them that were really interesting to us because they had been identified as restriction factors or other antiviral factors in the case of other viruses. So GIP4 activates the INF alpha mediated antiviral response in response to hepatitis C. ATG16L1 shows antiviral activity in INF gamma in mice and in vertical Zika transmission. EIF4G1 is cleaved in multiple picornoviral proteases, and TOC1 is required for function of IRF3, which is an antiviral transcription factor. So we were really curious about what was happening with these in Zika virus infection. So one of the things that we did was we looked at the ability of wild-type Zika virus protease to cleave these factors in human cells versus wild-type dengue. And we found that Zika virus protease was much better at cleaving EIF4G, ATG16L1, or GIP4 than was dengue virus protease. In fact, dengue doesn't really cleave, for example, GIP4 at all. But remember, we were interested in whether we could take advantage of this cofactor region. And so we made some hybrid cofactors, some hybrid proteases. So we made a version of Zika virus protease that has the core from Zika, but takes the cofactor from dengue. And when we made that substitution, we saw that we really diminished the ability of these substrates to be cleaved by this hybrid protease. We also made the opposite substitution where we took the cofactor from Zika and we put it on the core from dengue. And when we did that, we actually saw that we went from having, for example, no cleavage of GIP4 to pretty robust cleavage of GIP4. So just putting the cofactor onto dengue made it act more like Zika. So we said, this is really interesting. I wonder if we could really take advantage of this motion and make small molecule inhibitors that take advantage of this flexibility and these changes in conformation. So we wanted to take a chemical biology approach. We looked at the structures of the open conformation and the closed conformation, and we found that this residue here, alanine 125, sat right below the closed or the active conformation of these enzymes. So we asked, if we take A125 and we introduce a cysteine here, now we have a chemical handle. We can add a small molecule label of that cysteine and ask, does it impact function? And if it does impact function, that would be an idea that this mechanism would be good for controlling Zika virus protease in a therapeutic manner. So we did this experiment with wild type, and we added the small molecule chemical cysteine labeler, DTNB, had no effect on wild type activity, but in our A125C mutant, we saw full loss of activity. And if we added a reductant, DTT, so that we reduced off the DTNB, we gained back all of our activity. So we said, maybe we could develop some non-covalent small molecule inhibitors that take advantage of this open and closed ratio. So we set off to do that. So we teamed up with the UMass Medical Center small molecule screening facility, and we pulled out this hit, which we named it after the student who discovered it, Maureen Hill. So it's called MH1. And it's a two-lobed molecule that has an aminothiazolopyridine half and a benzfuran half. And the first thing we said when we looked at this was, we want to make sure that this doesn't have any non-drug-like properties. Taking a page from one of the previous speakers in this series, Brian Scheuket. And so we did a few tests to interrogate that. First, we wanted to make sure that it wasn't forming micelles or inhibiting in using a mechanism like that. So we added Triton 100, and we found that this had no impact on activity. So we said, okay, it doesn't look like it's a micelle former. We then changed the concentration because we said we want to make sure that it's inhibiting in a non-concentration dependent manner, so that the KD is really what's driving the potency that we observe, and we saw that that was true. We also wanted to make sure that there wasn't an aggregation-based mechanism, so we spun our samples and found no difference in the IC50. So this all suggested that these molecules are working in a manner that we might like. We then ordered some analogs of these compounds and tested their activity. And then Sparsh McKeck, a student who's sitting in the audience right now, made some more compounds. And what Sparsh discovered was that both pharmacophores, the aminothiazolopyridine and the benzfuran, were important for the function. And if we made changes to either side, we lost the potency that we had. So there's something about this particular compound that seems to work well for Zika virus protease. The next thing we wanted to ask is, does this compound work in a cell-based assay? So in this experiment, we treated cells with nothing or with the compound, and we saw that in the presence of the compound, then we saw complete blockage of cleavage of these factors, EIF4G, ATG16L1, and GIP4, suggesting that this compound is getting into cells and is effectively blocking the protease. So of course, at this point, we really wanted to know, where is this compound binding and exactly how is it doing its work? So Cristal Cruz worked hard to get crystals that showed us where this compound is binding, and that hasn't yet worked out. But in the meantime, she did some very interesting analysis using differential scanning fluorimetry. So if we look at Zika virus protease alone, we see that it has a melting temperature of about 43 degrees. Now, we wanted to know what happens if we bind an active site binder. And we hadn't developed one, but Kong's lab from Singapore had discovered this compound, compound 1, which covalently modifies the active site. This is the active site serine 135, and it leaves this little adduct on the catalytic serine. And when we added that compound, we saw a stabilization of 6 degrees, which is consistent with covalent binding. We then added our compound, which we had shown by mass spectrometry was not covalently binding, and we saw a similar stabilization of 7.2 degrees. So at this point, we started to ask, is this compound binding to the same site? Has a similar stabilization as if you bind this small fragment to the active site. But then we did the experiment where we added both the active site binder, compound 1, and our compound, MH1, and we actually saw additive stabilization, suggesting that these two compounds are not binding at the same site. They're not competing with one another. In fact, they're working cooperatively. So that additive thermal stability suggests that there's an allosteric site, an alternate site where these compounds were binding. So Cristal was really interested in pursuing this. And she said, my bet is that it's binding somewhere that's related to this cofactor region. So she took wild-type Zika virus protease, which we're showing in purple and yellow, and wild-type dengue virus protease, which we're showing in blue and orange, and she looked at the potency of MH1. She found that it's about 100-fold more potent against Zika than it is against dengue. And she then asked, what happens if I take different pieces of the cofactor region and substitute them. How does that affect? And she's going to look at both potency and thermal stability. So first she substituted the entire cofactor from dengue onto Zika, and she found that that decreased the potency somewhat. If she took just residues 50 to 60, which are residues that stabilize the cofactor and also increase solubility, these residues right down here at the base of the core region, then that also led to a decrease in the potency. So she also looked at residues 67 to 83, which are this blue dynamic cap that are at the top of the protein. And then she substituted just residues 74 to 86, which are a beta hairpin and make the wall that's negatively charged of this S2 region, which binds substrate. And finally, she took a region that's completely unstructured in every crystal structure that's been observed, which is residues 87 to 95. And what she found was that many of these had a deleterious effect on activity. And the most deleterious was residue 67 to 83. So this dynamic cap that's shown in blue here. She then wanted to ask, what happens to the stability? And she found that if she gets rid of 67 to 83 and substitutes those with the residues from dengue, now she loses the thermal stability that she had gained against Zika virus protease. So this really strongly suggests that this region is involved in the binding of MH1 to Zika virus protease. And if that's true, then the reverse experiments should also be true, which is if we take the dengue core and we substitute on the Zika cofactor, then we would expect that our compounds would actually bind better to that complex than they would to wild-type dengue virus protease. So when Cristal did the reverse experiment, she found that that was true. She found that when she substituted residue 67 to 83 from Zika into dengue, she actually gained about 10-fold potency. And additionally, she gained a little bit of thermal stability. So this suggests to us that these cofactor residues, 67 to 83, are important for increasing the sensitivity to MH1. They allow stabilization. But the fact that we needed the Zika core in order to get full potency and for full stabilization tells us that the Zika core is required for tight binding. And all this data together predicts a selectivity for Zika virus protease over other flaviviral proteases. So when we look at Zika versus dengue versus West Nile, we see that Zika is most potently inhibited by this compound. Dengue and West Nile are both less potently inhibited. So we next teamed up with a group from Utah State University, where there's an antiviral testing lab, and we convinced them to test our compound against all of their flaviviruses and ask how potently can the viruses how potently can our compound inhibit viral infection? And what we found was that most of these flaviviruses were not influenced strongly by MH1, but as our biochemical data suggests, Zika virus is inhibited by MH1. Now, that's interesting because in general, we would like to have insights in how to make a pan-flaviviral protease inhibitor. And what these data suggest is that we will really need to take into account what's going on with this cofactor if we want to make inhibitors that bind like MH1 does. Now we have some more data that is really helping us to understand what's going on with this region of the protein. And this data came from NMR. So the experiment that we did here was we looked at Zika virus protease alone, which is the blue spectrum, or in the presence of MH1. And when we first saw this, we said, oh, this is odd. This looks like we're really losing some of our signal. And so we decided that it's impossible at this point to tell whether it's a problem that would be something like dilution, or is it a problem of a change in the dynamics. And a way that you can measure this is by changing the field strength by which you do this experiment. And so we did the same experiment at NMR FAM in Madison, Wisconsin on their 900 megahertz spectrometer. And what we saw there is that when we bound MH1, we didn't lose any of our peaks. And so it tells us that there's an intermediate exchange dynamic effect in the presence of MH1. So we can see all those resonances at 900 megahertz, but we can't see them at 600 megahertz. So what we're now doing is we're looking at this compound, MH1, in the presence of wild-type Zika or the Zika hybrid. And when we look at this hybrid, which binds MH1 more poorly, we see that we have similar dynamics with and without the compound, whereas in the wild-type enzyme, we've altered the dynamics significantly in the presence of the compound. So the hybrid dynamics are less impacted by MH1, and this suggests that the NS2B region, this cofactor region, is more like the MH1 bound state. So the dynamic of this region and likely other flaviviral proteases are distinct from Zika virus protease. All right, I think I have just 2 or 3 minutes left. And so in that time, I will just give you a very short preview of some of the work that we're doing on chikungunya virus protease. So again, this audience probably needs no introduction to chikungunya virus, but the virus itself has a protease that's called NSP2, and it's a protease of 38 kilodaltons that has a catalytic cysteine histidine dyad. So we decided that we would like to make inhibitors for this protease, and there were a few things standing in our way. One of the things was starting chemical matter, and another thing was that there isn't a very good substrate available for chikungunya virus protease. So Sparsh Makek has done some experiments where she's taken chikungunya virus protease, and she has screened using fluorine-19 NMR, looking for small molecule binders, not necessarily inhibitors, but binders of chikungunya virus protease. And what I'm showing you here on the left is one of her hits, which had a KD of about 7 millimolar, which is what we're expecting. We're looking for binders, not necessarily for tight binders. We then ordered an analog, and that analog has a slightly more potent binding. And we can see that binding as evidenced by a chemical shift perturbation as a function of concentration of this binder. So we've been able to make a really lovely dose response, and we can measure these KDs very accurately. So we're excited about these fragments for a couple of reasons. One is these fragments are starting positions for developing inhibitors that bind in different ways, or that can be linked up in new ways, potentially with active site compounds, or potentially using a PROTAC approach. The other thing that we have going on in the lab is that we've also assigned 50% of the residues within the structure of chikungunya virus protease, so that we will be able to identify the site of binding of these compounds. And then the last thing that we've done that may be of interest to people in these groups is that the existing substrate that's been published in the literature was really had very low signal to noise. It had a Dabsel-Edans pair. And when we did a pilot screen, we found that the signal to noise in this screen was about 2, was actually worse than 2. And so we weren't able to do any screening. We've now teamed up with Marcin Drag and in Warsaw and have developed a new substrate for chikungunya virus protease that now gives us the kind of signal to noise that will allow us to do the kind of screening that we're interested in. So I'll end up there telling you that we've made an inhibitor that targets the NS2B, NS3 interface. We've got evidence the dynamics in the NS2B region are key to distinguishing the activity of Zika virus protease from other flaviviral proteases. And even if we use an active site inhibitor, I think that dynamics contribute strongly to the activity and should be considered when you're developing a pan-flaviviral inhibitor. We have ongoing NMR work on both our chikungunya and our Zika virus protease targets. And I'll just sum up by thanking Christal, Sparsh, and Maureen, who are the 3 students who did the work here. And I'm happy to stop there and participate in the question session.